Okay, so let's get going on our next group of plants. So this week we're going to be looking at the gymnosperms. Okay, so recap of where we are. Right, so from last time, we saw that we had three major groups of vascular plants. So we started with our mosses, our hornworts, our liverworts, our bryophytes, and then after we developed vascular tissue, so xylem and phloem, allowing water, sugars to travel around the plant, we then group our plants into three divisions. So the pteridophytes were the ferns and fern allies that we talked about last time, reproducing by spores. And now we're going to move into our groups of plants that have seeds. So where we are right now is we're starting with the division Gymnophyta. So these are going to be our gymnosperms. So they're going to have seeds, but what they're not going to have are structures called flowers. Okay? So our flowers will be found in our angiosperms later on. So we will be focusing mainly on that after spring break. But we are starting to see plants that use seeds rather than spores for reproduction. So on our timeline, we developed vasculature after we developed land plants. Now we have our first seed plants. So we can see that this split is much younger, where we have our gymnosperms and angiosperms splitting off from each other. And we are located right here for today. All right. So when we're talking about the gymnosperms, we're going to have seeds. So we will also sometimes refer to them as spermatophytes. That is a word that refers to both our gymnosperms and angiosperms because they both have seeds. So that would be like group B at the bottom here. Within our gymnosperms, depending exactly on our phylogeny, which one we're looking at, which paper we're looking at, we're going to have four probably classes of plants that we're going to talk about. So we're going to talk about Chenopsida, which is really a conifer. We're going to talk about Ginkgoopsida, which is really just kind of one genus of plants, the ginkgo. Cycadopsida, which are cycads, and then Gnetopsida, or sometimes referred to as Gnete, which is its own weird little group. Okay, so when we see phylogenies vary, that Gnetopsida is going to be one of the ones that is particularly confusing. But we'll look at that in a little bit. Okay. So we want to start out by understanding what a seed is. So presumably from everyday life, we can kind of begin to think about what we would call a seed, right? We planted some seeds in the back. You eat an apple or an orange, you find a seed inside. What that is, botanically, is a little embryo. So it's a little plant embryo enclosed in a protective outer coating that's going to protect it from drying out. The cool thing about having your plant embryo in a seed is it can be kind of dormant and travel around for a long time before it starts to grow into the adult plant. Okay. The word gymnosperm means naked seed. So what this means is that our seed isn't surrounded by an ovary. When we look at our angiosperms, our flowering plants, we're going to see that they have fleshy structures forming around them, descending from the plant's ovary. And that's really what the fruit is. So our gymnosperms don't have that. Instead, we're going to see that our seeds are usually going to be contained in like cones, but they're going to be exposed to the elements. So if we're comparing a seed to a spore, we have a little Venn diagram down here on the bottom. So major difference is that a seed has a multicellular embryo inside it, whereas a spore is just a single cell. Okay. And a seed is also going to have a supply of nutrients for the growing plant for when the seed sprouts. Not true for a spore. Both contain genetic material from the parent plant. Both do have some protection from dry conditions because we are talking about land plants, some forms of protective coating like we saw on our ferns, but these will look different depending on the specific plant we're looking at. So just a tidbit about the word germination. So when we talk about germination, what we're talking about is the growth of a new plant. So we could use the word germination when we're talking about a spore. So the new plant, the sporophyte generation growing up from our fertilized spores, so single cells that came together when we were looking at our fern life cycle. But usually when you see the word germination, you're looking at like a packet of seeds that you're planting in a garden. So what we're talking about then is the new plant growing out of the seed being an embryo already. So those embryos inside the seed can last a long time. They have seed banks where we're preserving seeds for future generations. They've grown seeds from thousands of years ago, honestly. You can revive plants. Sometimes those seeds are still good for a long time. Coconuts are seeds that last for a particularly long time. They float around in the ocean until they find new islands sometimes. So you can see a picture of that. Seeds in general kind of have more ways to travel due to their size, their protection, and that supply of nutrients. Spores are going to travel mainly by the wind, kind of locally, or in the cases of our mosses, through the moisture. But when we have a seed, that embryo in its protective coating, larger, it's going to end up spread out over potentially quite long distances. So sometimes they're going to spread by the wind, but they're also sometimes going to be carried by birds, insects, and other animals as they get kind of carried around, often through an animal eating the seed and then pooping it out somewhere else after the animal itself travels. So when the seed germinates, it got to where it's going, got to some new place, some distance away from the parent plant. That seed is going to start to absorb water from the soil it lands in. So the embryo is absorbing water. And then there are nutrients stored inside the seed that feed the growing plant for a little while. Which is why we can see, right, that coconut already sprouting up even while it's in this water, right? Because it doesn't need to get nutrients from somewhere else. At least not at first. Okay. So in terms of how our gymnosperms split off, how they evolved, what we first see in the fossil record is a plant that looks kind of like a fern, but instead of having spores and sporangia on its leaves, it actually literally has seeds on the leaves and some on the branches as well. So these plants weren't really ferns, they just kind of looked like them. We refer to them as seed ferns or Gymnosperms. They don't exist anymore, but we can see them. We can see that they're actually quite large sometimes when we find them. And here's a reconstruction of what this might have looked like when it was alive. So you can see the little brown seeds here growing directly off those leaves, kind of in the position where we see the sporangia in some of our ferns. So we're moving from these seeds directly on leaves to some really characteristic reproductive structures that we see in gymnosperms. The word extant just means things that are alive now, but not fossils. So these seeds that we see forming in our gymnosperms are going to be contained within cones usually. So we have some pictures of typical pine cones that you might have seen outside. These are actually specifically female cones, specifically of some of our species in the Panopsida. There are also male cones, which look fairly different. So here we see a pollen cone from some of these same species. So in lab on Thursday, we'll be looking at different types of cones, how we can describe them, how we can identify them, and a little bit about their kind of life cycles and maturation. Okay. So these cones that we see might look a little bit familiar, and sometimes you will see them referred to as strobili. So that's because they are a similar kind of structure to what we saw in our lycophytes, our genus Lycopodium. So when we were looking on the slides for, oh wait, the wheels go. Okay. Okay. So when we were looking on the microscope slides or on the tables at Strobeli, we saw they had this kind of structure, which was a little bit cone-like. So they had these little leaves, and then they had their spores, male and female, that sort of nestled in among those leaves. So in this lycopodium, we can see here that we have a spore-bearing leaf, our sporophyll, and then we have the spores nestled in here. Okay, so this is similar to the structure of our cones of our gymnosperms, but our difference is that we're now going to have the formation of seeds rather than just spores. And when we look at gymnosperm cones, they're also going to be larger, and they're often going to be woody, like we see in the pine cones over here. And whereas when we were looking at our club mosses or lycopods, we had these little green kind of subtle fleshy structures. So we're going to look at a couple of different types of cones as we look at our groups. So we're going to start with our conifers. So you've seen some conifers actually in our first lab when we were looking at just getting our head around how to use dichotomous keys, how to identify plants. So we were actually looking at the conifer section of a Minnesota trees book to practice that. So we'll see a bunch of those same plants again on Thursday. And as we think about this, now we're just going to dive deeper into their biology and kind of expand on the groups we're looking at to sort of expand our knowledge now that we know some things about this group. Okay. So just a reminder of some of our terms that we've talked about so far. So remember, our sporophyte is our diploid generation of a plant. Plants have alternate generations, haploid and diploid. So when we're looking at gymnosperms, you see a tree, that's the sporophyte generation. Gymnosperms are going to be heterosporous, so they're going to produce both male and female spores before they produce those seeds. Okay, so we want to remember some words related to heterosporous plants, so to male and female spores. So a sporophyll is like a modified leaf thing that can produce spores. So our microsporophylls and our microspores were male structures. So they produce male spores on a spore-bearing leaf that is then going to have these sort of smaller male spores that create sperm that pollinate the female structures. When we talk about this in our larger plants, our male spores, we're going to come to refer to as pollen. Pollen is basically male. So our mega sporophylls, our mega is larger. So the larger structures are going to be female spores and female spore-bearing leaves. Okay. So those led to female gametophytes when we were looking at our ferns, essentially. And then our strobilus was that cone-like structure, and now we're going to be thinking about like pine cones, so actual cones. Okay. In the definition for strobilus, referring to it as a mass, so a group of scales or bracts. So just an FYI, a scale is going to be like a dry little leafy structure. So basically, the scales on a pine cone are what you would expect them to be, those pieces that you can pull off. Bracts are related, but you're going to see different types of bracts kind of throughout this course. So a bract is just going to be a modified leaf or a modified scale. So it's going to be some other kind of additional structure taken out, essentially. So we will see some pine cones that are going to have both scales and bracts, which is why I'm mentioning this now. Okay. All right. So these male and female structures, if we're looking at our conifers, so we have typical pine cones and pine pollen here as our example to begin with, all right? So we're going to refer to our typical woody pine cones as ovulate cones. So they're ovulate cones because they are going to have little ovules at the base of their scales, okay? And that ovule going to get fertilized and it's going to form our seed. So this is where we're going to see that developing plant embryo sticking here in this ovulate cone. So these structures are all female. And you can see a section under a microscope here as well of what that looks like. Okay. Actually, here we can kind of see the difference between our bracts and our seed scales. So the scales are the ones that have the seed sitting on them in this example. And we'll take a look at these structures in that. Okay. So this is the female structure. These are essentially our eggs that are forming the embryos. On the other side, we have our pollen cones. Okay. So our pollen cones are therefore made up of essentially microsporophylls with our pollen, which is just microspores, microspore mother cells that are then going to disperse out into the world and pollinate our ovules on the female cone. So these are going to, in general, look kind of fleshier. They might be smaller. They'll look different. They're not going to be our typical woody pine cone. Now, when we have I'll back up a second. So we have two different types of cones, different types of structures, right? We have our male pollen cone and our female ovulate cone. When we're referring to an individual plant, it may be the case that, say, a tree has both typical ovulate pine cones and also male pollen cones, or it may be the case that we have a male tree and a female tree. So we have different words for that. So if we have a male tree and a female tree, so like two types of tree, we're going to call it dioecious or dioecious. Okay. If we have a tree that has both of them on the same cone, we're going to refer to that as monoecious because they're on one single plant, right? So every individual plant has both. So every individual plant is kind of the same. So we just have like one type. In the case of a monoecious plant, you will see often a different sort of arrangement between where you find the male pollen cones and where you find the female ovulate cones. So usually the pollen cones will be up higher so that when those spores get released, that pollen gets released, it kind of falls down, right? So they'll be able to kind of self-pollinate potentially, or at least have that pollen go farther, right? Because they start up higher. So when we're thinking about our life cycle here of a gymnosperm, let's start. Yeah, okay. We're going to start here with our typical tree. So our typical tree is a mature sporophyte. Okay, so here we have our mature sporophyte, and it's going to be making its different types of cones. So this is going towards both sides. All right, so we're going to pretend this is a monoecious mature sporophyte. So it's making both male and female structures. Okay. So on the female side, it's making a woody ovulate cone here. And if we look at a scale, it's rather large, and we would find our little ovule at the base of it. In our pollen cone, we have our microsporophylls with their microspores, aka pollen. All right, so we see that there. Okay. So this is right before we develop the pollen. Okay, so we have a mother cell that then divides twice by meiosis, creating our gametophyte. So the pollen here. Okay, and the same thing is happening in the ovulate cone. We just have a larger structure, right? So that megaspore, right, is forming the female gametophyte via meiosis over here. We can see that we will continue to use some of the other terminology that we were using for ferns. So we're again going to call the female structure here an archegonium, okay, on the female side, and our male gametophyte is going to have to get over to the archegonium to that female gametophyte to fertilize it. So it's going to have various structures that help it move around, potentially wings. It's going to have cells that are dividing cells, generative cells, as well as tube cells, which are going to kind of create a pollen tube to go down into the archegonium. Okay, So fertilization occurs, and then the embryo starts to develop. So the embryo is developing here where we saw the archegonium in this structure that was at the base of the scale was the megaspore and the female neophyte. So we can see that developing embryo here. And ultimately, this is what forms the seed. Okay, so the seed will often have a larger wing. You may have seen these sort of spinning around out in the wild or floating on the wind. Okay? And that seed is going to land on the ground somewhere, going to encounter moisture, going to germinate, and we're going to have our little tree spring up and form our mature sporophyte. So this is our life cycle. Okay. So let's talk about a couple of examples of species or genera within our conifers. Okay. So at the moment, at this current point in time, our Gymnosperms or conifers are our most abundant group of gymnosperms. They're in about 8 families, so actually quite manageable compared to our angiosperms. And all of them are going to be woody plants. They're mostly trees. So our woody plants are going to stay around year after year, right? They will lose their leaves, but when we're talking about our conifers, many of them are evergreen, which means they're not losing them all at once in the fall, at least not usually. They're losing them kind of one by one, the way you lose individual hairs from your head. Lots of our conifers have something called resin. So resin is a substance that the tree is producing to protect itself against infection. So if you've seen, like, you cut a branch off a pine tree or something, you see that oozy stuff that starts to crystallize. That's the resin, helping the plant kind of heal over and not get an infection through that wound. And we know some stuff about the past because resin fossilizes as amber. So some of the things we know about the ancient past are because we got things stuck in that sticky resin. We actually see it still around preserved today. The Gymnosperms are pretty cool because they have some sort of like Guinness Book of World Records type of trees. Our tallest trees, our widest trees, our oldest trees are pretty much all within this group. So these are our conifers. So we can see one example of a phylogeny of gymnosperms here. Okay, so we have our gymnosperms splitting out. So over here we have groups we didn't talk about yet, our cycads and our ginkgos. And in this example, our conifers are splitting out with our gnetophytes and then angiosperms out to the side. Okay. So in this diagram, we might say that the conifers and the gnetophytes are depicted as a sister group. Ginkgo and cycads are splitting out separately here. And then the gymnosperms and angiosperms are also sister groups here. Okay, so a couple families. So one of the families that we looked at was the family Pinaceae. Maybe we didn't know this, but when people talk about pine trees, they're usually referring to this whole group, but actually only one of the genera within this group actually called a true pine. So our true pines are really in the genus Pinus, but this whole family has plants that look kind of similar. So we have pine trees, our true pine trees, but we're also going to have our firs and our spruces here. So these you'll all see pretty commonly outside around us here. We have plenty of conifers. So if you're trying to tell the difference between whether you have like a real pine tree, a true pine tree, or you have a spruce or a fir, there's some things you can look at. So when you're looking at pine trees, you're going to see that you have relatively long, relatively soft needles that are going to be in bunches. So you're looking for that bunching structure, so little bundles as they join to our twig, right? So here you can see those bundles of needles, okay? And they're attaching in those clusters. So you can rule out pine pretty quickly or rule it in just by looking at that. Spruces and firs are a little harder to tell apart. Some of the differences we're going to see is little bit on hardness. The spruce tend to have harder needles. The firs tend to have softer needles. But what's going to be really noticeable is the shape of that needle kind of in cross section. So what I like to do is pick a needle and then try to roll it between my fingers. So when we say that the needles are four-sided in a spruce, what we're saying is, so like, here's our twig, here's our needle coming off the side. We're saying that if we slice it like this, you'll get a cross section that's kind of squarish. Okay. Versus if I were to slice here across one of these fir needles, I'd end up with something kind of flat. Okay. So the reason I like to roll them between my fingers is that something with multiple sides like this is going to roll much better than something flat does. And that's kind of what I'm trying to feel for intuitively. The growth pattern, though, of those needles will also differ some, usually. So in a spruce, the needles are going to be kind of all around the branch or all around the space. Whereas on the firs, you're going to see them kind of flat. So you're going to see like branches out one side, or sorry, needles out to one side, needles out to the other. But you're not going to usually see them going like up and down. So take a look at some of our very tall, very old, very wide conifers. So our tallest tree is the coast redwood, the Sequoia sempervirens. So they've actually named the tallest trees. People go out into the woods and they try to find the tallest tree, and they name some of the really, really special trees. So the current tallest tree is a tree known as Hyperion, and they won't tell us where it is. So we try to protect these really ancient, really tall, really extreme plants and trees because they took a long time to grow. If someone goes out and cuts it down or harms it, right, it's one individual tree. We're not getting that back. So its location is secret. It's thought to be 600 to 800 years old or so, so quite old. 379 feet tall, at least the last I looked it up. Plants grow, so maybe it's taller than that now. And we can see it compared to some other plants here. So a typical oak tree like this, about 35 meters tall. Here we have Big Ben, right? And we can see that we have two known trees that we know the names of, or that we have named, that are taller than Big Ben. So another one is Centurion. So that's a mountain ash, but our Sequoia Hyperion is the tallest that we know of so far. So I think it's somewhere in Redwood National Park, California. So location being secret is like the exact location, right? They don't want a bunch of people hiking out to it, touching it, messing it up. Now, if we say a tree is the biggest, we have to specify what we mean because it's not necessarily the tallest, right? Big can mean multiple things. But our widest tree, which is another measure of size, is also a conifer. So it's a tree called the Montezuma cypress, Taxodium mucronatum. So the local legend where it is that it was planted 1,400 years ago by an Aztec priest called Pichocha. As far as scientific estimates go, that's somewhat realistic. Estimates range pretty widely, but we think it's like 1,200 to 6,000 years old. So not super specific. We're not going to cut it down to see how many rings it has. So we'll see. And it's 138-ish feet in diameter, known as the Arbol de Thule. If we were thinking about biggest, we might also think about volume, right? So things are tall, things are wide. You can also measure it all together and get a volume. So our biggest tree by volume is the giant sequoia, so Sequoiadendron giganteum. This one is thought to be 2,200 to 2,700 years old. We can see that this one you can get to. We can see that we've taken some measures to protect it. So a fence to stop people from really messing with it. It is in an area that does experience wildfires. So here you can see some workers wrapping it in aluminum foil to try and prevent it from burning down. So we are taking good care of this giant sequoia. Now, there are other giant sequoias. You can find them in Sequoia National Park. You can also find them in Yosemite. You can find them out west in general. I think it's super fun that there are some that they have literally built tunnels through. So I have listed their locations for you in case you want to plan a tree-themed vacation. So the tunnel log is in Sequoia National Park, and Tuolumne Grove is in Yosemite. And you can see how you can get through. Well, I don't think they let you drive through this one in a car anymore, but you can see how large this archway that they've created by cutting open the tree is. I think this one is Tuolumne fallen over, but they've made an archway here as well. But this is also part of why we're keeping secret the location of some of our really big trees, right? You can see that people had fun, but kind of dangerous for the tree for some of these. Our current oldest tree, we believe, is Methuselah, which is a Great Basin bristle cone pine, Pinus longeva. Right, so from dating, it's confirmed to be almost 5,000 years old. The estimated germination is 2833 BC. You can find it in Inyo National Forest, somewhere in California or Nevada. Exactly where it is a secret. There used to be an older tree, confirmed older. Yeah, you can see its ages on there, 4,862, 5,000 years old. A grad student was out with the Forest Service working in the National Forest and accidentally cut it down. So that is another reason that we are keeping the location of some of these secret. I feel bad for the tree, but also very bad for that grad student who didn't know what they were doing. Another fun tree story that I always really like. So the Dawn redwood is a plant formerly known as Metasequoia glyptostroboides. So 65 million years ago, it was kind of everywhere. It covered North America. It's cool as a conifer, partially because it's deciduous. I don't think it's the only deciduous conifer, but it's a prominent example, and it's not true of most of our conifers, which are evergreen. So you can see how it changes color in the fall, and then it will lose its leaves. So back in the day, they found plenty of fossils of this thing, but they thought it was extinct. Then they found a tree in a little valley off in China. So it took people kind of a while to realize that they had found a living example of this tree that we thought was just very, very ancient and extinct. But you can now see it out in a lot of botanic gardens, a lot of arboretums, because after they found it in this valley in China, they redistributed it around the world. So they gave it out as gifts, sent it to other botanical gardens, arboretums, plant places, universities. So now we can see many examples outside for us to view. And you can pretend you're a dinosaur running around below this tree. So now we're going to walk through some of our other groups of gymnosperms. Okay, so we were talking about the conifers. So this would be the pines, and the redwoods may or may not be a separate group, again, depending on what phylogeny we're looking at. All right. So now we're going to talk about our cycads and ginkgo, which may or may not be sister groups to each other. Okay. So we're going to start with the cycad. So the cycad looks like a palm tree. It is actually not a palm tree. So I probably shouldn't have shown you that picture of a coconut earlier, because a coconut is not a gymnosperm. It's an angiosperm. It's a monocot, but it's cool, and it's a cool example of seed dispersal. However, our cycads, they are gymnosperms. So they're going to look like they have a trunk. They're going to look like they have kind of fronds. So these are the leaves of the cycad here. And then in the center, they're going to have their cones. So these plants are actually at risk for extinction, and there are some crazy stories you can find in newspapers about people smuggling these plants. The highest price I found for one was $50,000 for a illegal cycad. There are a couple you can also find in garden centers though. So they used to be a lot more prevalent. They are not doing so well right now. And they are part of our plant poaching trade. Some cool fun facts about our cycads is that Zamia rosellii is an example of a cycad, and if you looked at its sperm, you would notice that it had 40,000 flagella, so little whip-like tails from it. Most of our plant sperms don't really have flagella at all, so Zamia is really extra here. Okay. Another cool thing about cycads is that they have a symbiosis with a bacterium. So cyanobacteria are a type of blue bacteria, really. One of the things this enables them to do is that they can fix nitrogen. So one of the things that plants sometimes do have trouble getting for their growth is nitrogen. A certain limited number of plants are able to form different types of symbioses to let them make the nitrogen like in our environment bioavailable to them. But most of our plants can't do that. So that's part of why we have to put down like fertilizers and stuff, is that a lot of our plants are unable to use the nitrogen that is, for example, in them. But our cycads can do it. Okay. So here we have some more examples of cones, some weirder looking cones that I think are fun. So I can see some similarities to our pine cones, but we can also see some notable differences. I really like this picture of a cycad female sort of ovulate cone because its ovules are super duper obvious. Okay, so we can see these green structures on this female cone-like structure. Okay, so those are ovules. So we would call these leaves or scales with those ovules megasporophylls. Okay. We also have a separate male structure. So on this structure, this is going to be analogous to our pollen cones in the pine, but you can see it is bigger, woodier here than we saw in the pines. So it does have these sort of scales that we can see, which we might call microsporophylls. And if we zoom in way, way, way down, we can see that size difference on those microspores. So you can see big difference between those ovules, those megaspores in the female structure, and those tiny little microspores when we're looking at the male structure. Something you may or may not have noticed as a difference when you're looking at the microsporophylls and megasporophylls in lab last week. So that kind of distinguishing size characteristic kind of continues through here. Okay. So we're going to move down to our possibly sister group, the ginkgos. So there's actually only one species in one genus remaining in the world. And as far as we can tell, it has not been evolving, not been mutating for about 200 million years, which is kind of crazy. Doesn't seem like that should be true, but that's what we got for now. So ginkgo biloba is going to have these leaves with 2 lobes. That's where it gets its species name. So 1, 2. Okay. It is dioecious, so that means that it has separate male trees and female trees. Fun fact, because these leaves are pretty, they turn a pretty yellow and fall in the fall. They're a pretty commonly planted street tree. So they can live hundreds of years, so you don't need to do much maintenance. You don't need to worry about these trees dying, replacing them. That's very convenient. What is inconvenient about them is that the female trees smell absolutely disgusting. Okay. So the female trees have really, really, really horrible smelling berries or cones. They've been described as dirty gym socks, vomit, or poop smell. And I can corroborate that. There was a female ginkgo tree around the corner from my apartment in grad school. I definitely thought I stepped in dog poop like every day for a couple of weeks. But It was a female ginkgo tree. Okay. So the problem here is that urban planners are not totally stupid, right? They're like, okay, it's fine. It's only the female tree that smells horrible. We'll just plant male trees. It'll be fine. Okay, which they do. So they take mature trees that they can tell what sex they are. They take branches and they graft them onto rootstock, basically making clones of the original tree. So that works fine, except for ginkgos can do a cool thing, which is that rarely they can spontaneously change sex. So sometimes they plant all male trees down the street. Everything's going good for years and years. And then all of a sudden, one of the branches decides to be female and starts creating these horrible smelling berries. I think that's kind of fun and tricky of the ginkgo. I don't totally mind. The berries are actually also edible. So if you can stand the smell, you can go collect them and dry them. And apparently they taste pretty nice. So if you find any female ginkgo trees around, let me know. I've never tasted one. I'm kind of curious. Okay. So when we're thinking about their reproduction, or if we were trying to tell whether we had a male tree or a female tree, we'd be looking for its reproductive structures. So it's cones. The male ones look pretty cone-like. They look kind of like our pollen cones did on our typical pine tree. So this is a male ginkgo tree. The female cones look weird. So the female cones don't really look like cones at all. You have like, let me go trace this right. So you have a little stalk kind of structure and then just two little ovules, okay? Which then as they develop, form a larger structure like this that does have a little bit of a fleshy coating around it. So these are not technically a fruit. Fruits are specific to our angiosperms because the ginkgo will actually form the structure regardless whether it's fertilized or not. So it has like a little bit of a different embryonic origin. It's going to look like fruit. So if you're looking at these structures that turn kind of yellow and then drop off, you got a female ginkgo tree. Okay. So for our last group, I want to talk for a second about wood, because that's one of the things that's going to be weird about one of our examples. So we've been talking about, we've mentioned xylem and phloem, right? So that's our vascular tissue in our vascular plant. So we had some in the ferns. We didn't zoom in too deep on them, but the xylem carrying water, phloem carrying like sugars and our products of photosynthesis. Now, if we're thinking about wood, so long-term tissue that's getting old, that's staying around year after year, we're going to kind of replace some of that xylem and phloem. So over time, some of our xylem is going to become lignified. So this means it's picking up a molecule that kind of stiffens it, and it's losing its ability to actively transport water. So that's what we can see happening in the center of like a tree branch or trunk. So the centerpiece is essentially old xylem. So this old part of the tree we're going to call the heartwood if we were to take a cross section through the tree. We do still need to transport water, so we are going to have new growing wood. So the next layer out we would refer to as the sapwood. So here we have our sapwood. Right. So this is where our active transport is going on. Okay. And then we'll have further layers kind of protecting. So we have a soft layer called the cambrium here. We have a layer of phloem right there. And then we have the bark, which is actually old phloem that's kind of getting pushed out to the edges. Right. So this progression of growth over time is what creates rings. So if you've ever looked at a block of wood cut like this and tried to count rings to figure out how old a tree is, this is what you were doing. You were counting basically years of growth of the xylem. Okay, so we can see kind of lines where we had this kind of transition from old xylem to new xylem. Now, if we were to zoom in on some specific wood, we would see, we went out to like Menards or something, took some planks, slapped them under a microscope, we would see probably one of two kinds of structure. So In our softwoods, which are going to be our gymnosperm woods, so you get a plank of a pine or something, you're going to see lots of little holes, almost, we're not going to say randomly dispersed, but all over the place, right? Just lots and lots of little holes. Right? This is part of why we call them softwoods. There are some gymnosperms that are very hard. There are some hardwoods that are very soft. But because of this structure, this kind of loose structure, on average, our gymnosperms tend to have a softer texture of wood. It's different than our hardwoods, which are going to be really packed together, right? So you don't see all these little holes, right? You see everything really packed together, and then you see some really big holes, okay? So that's going to be wood that comes from one of our flowering plants, one of our angiosperms. So the reason we see these differences is that we have different sort of vascular structures. We have different kind of types of structures in our xylem. So in our gymnosperms, we have a more primitive or earlier evolving structure that we a tracheid. Okay, so these are these structures that we see here, kind of in blocks, right? So we're looking at the xylem here. And if we were to look at actual cells, they're kind of tapered and pointy. They're kind of interspersed, a little more like this. Okay, so that's what we see in a gymnosperm. Versus in an angiosperm, the way we're making those big holes is we have these longer, bigger, wider structures called vessel elements. Okay. So that's what we're looking at when we're looking at those big holes. And then the wood surrounding is really just made of kind of like hardened elements rather than open passageways for water. Okay, so that's what's going on there. So we can see those tracheids interspersed just kind of everywhere throughout our softwood, our gymnosperm wood. Right. And the reason I want to mention that is that one of our groups in the Gymnopsida or Gymnopers, or however we're going to classify these plants, Gymnophytes, right? Depending on exactly what our phylogeny looks like. One of the examples in the plant is going to have structures in its wood that look a little bit more like the angiosperms than our gymnosperms, which is where it is currently classified. So the Gymnopsida, their classification is kind of unclear. If you were to look at the different species that are within this group, they don't really have many synapomorphies. So remember, an apomorphy is like a derived characteristic, so like a new mutation. A synapomorphy is an apomorphy that you see in the whole group. Okay, so they don't have many common characteristics other than ones that we assume they inherited from the last common ancestor of the other plants. Okay. So we can see here that in one of these examples, we have the Gymnophytes as a sister group to our conifers, right? And then the cycads and the ginkgos doing their own thing at potentially like a three-way split. Another possible arrangement of these is that the Gymnophytes are actually a sister group to the angiosperms. So they're not flowering plants, but a sister group of the flowering plants, and not really within this separate conifer group at all. Sorry, a gymnosperm group at all. I think that one's less likely. Another option is that we have them coming out just separately from the pines, nested somewhere within the conifers. Another option is that they're just an outgroup to everything, doing their own thing. So this like grid of images comes from Wikipedia, but they all drew these from different papers arguing about how exactly we should classify this group. So we see some examples on the right here, and we'll take a look. So first one I'm going to mention, because this is why I was talking to you about wood, is Needham. So this is a group of trees, shrubs, and lianas lianas are like big, thick vines that you'll find in rainforests that look a lot like angiosperms, even though we think that they are gymnosperms. And one of the ways in which they quote unquote look like angiosperms is they do have those sessile elements in the xylem rather than just the trachea. So if you were to take a little cross section through their wood, right? So if you were to look in that wood, you'd see kind of that hardwood seeming structure. Another thing that they sometimes do is something called double fertilization, and we'll talk about that when we get to the angiosperm. But it's a specific part of the angiosperm life cycle and reproduction. So those are questionable things about those. Now we're going to be looking at some individual genera as examples. Okay. So the first one we're going to think about is ephedra. So ephedra is cool because we've taken some drugs by isolating components that this plant makes naturally. So ephedrine is derived from ephedra, and pseudoephedrine also kind of a form of this same drug. So pseudoephedrine, like where you get the name like Sudafed, right? This is a decongestant. What it does is kind of activates your sort of sympathetic nervous system a bit more, helps you lose congestion as it does this. And they used to make diet pills out of ephedrine, and then they banned them. Okay. They made great diet pills because it increased the metabolic rate, right? So you burn calories faster. Unfortunately, at the same time, ephedrine was causing potentially heart attacks, potentially psychiatric episodes. So the FDA decided that this was not great. So ephedrine supplements were banned. However, people have been using ephedra to treat illnesses back to like 5000 BC. So this does have a long history of kind of like local medicine where this plant grows. So presumably you could avoid heart attacks with dosages, or maybe not everybody cared all the time. Who knows? People do some dangerous things. Another plant that does not look at all like the other plants in this group is Welwitschia. So some people call this the world's ugliest plant. So that's another sort of Guinness Book of World Records sort of plant within our gymnosperm group. It does look pretty ugly, but I think that kind of makes it cool. So these plants can also live a really long time. Some of the individuals that we've found, we think are over 1500 years old. That is a picture of one up here in the right. Crazy thing about this is actually only 2 leaves. I don't know exactly where the split is, like maybe there or something. Because this plant gets so old, has only 2 leaves, has wind constantly buffeting it, the leaf just keeps growing and getting split and split and split. So these are just like little chunks of one leaf that got split up as this plant grew. It is evergreen, so it's not losing those leaves. It's found primarily in Namibia and Angola. So it is under threat a bit. The plants in Angola are actually better off for a sad reason. The reason being that there are lots of landmines where you find them. So people are not going out and poaching Welwitschia because they do not want to step on the landmine. Interesting facts here. As we're naming plants, there are some controversies in naming systems. Historically, lots of plants have just been named after whatever European explorer first saw it, rather than things about the plant itself, things about how it's used locally. And there's some sort of evolving ideas about how we should be naming plants. But the European guy who first encountered this actually proposed a local name for the plant itself, proposed calling it Chimboa. But a British botanist decided that he should just call it after the Austrian guy anyway, despite the fact that that was not the proposed name. Okay. I want to watch a little video about this world's ugliest plant, and this is about where we're going to finish off. Volume. How we do?
As is the case the world over, the physical adaptations of the flora and fauna are often representative of the environment they inhabit. We have learned that many arid adapted plants have lost the large leaves of their temperate cousins. However, there is a plant endemic to the moment that defies the ordinary. It is so unusual, yet perfectly adapted. Welwitschia mirabilis, a true living fossil. The interesting thing about Welwitschia is that the leaves seem to break the rules for what we would predict leaf sizes of plants and an environment like this to be. So there's quite a bit of work been done to predict the relationship between environment and leaf size. And one of the conclusions from that work is that if you have a plant that's in a hot, sunny environment, that plant should produce leaves that are small, numerous, and narrow. And here's Welwitschia, it breaks the rule. These large leaves, when you're dealing with a healthy and intact plant, provides an enormous patch of shade, shelter underneath it. And this is a phenomenon that's called niche construction. It's an organism actually building an environment in another environment. So you have these patches of shadiness and relatively cool temperatures under the canopy of the Welwitschia leaf. And this might not only help Welwitschia, for example, cooler temperatures, higher humidity, there's less demand for water on these plants to do what they need to do, but it can also provide a shelter for other organisms that live around the Welwitschia. And this is a relatively unexplored area, and that's, in part it's because niche construction is a relatively new way of thinking about adaptation and evolution, and this may be why Welwitschia leaves break the rule. The evolution of the angiosperms, the flowering plants, took place at the beginning of the Mesozoic period, some 250 million years ago. This period was when the amphibian dominance of the Earth started to fade and the reptiles began to emerge. About a third of the way through the Mesozoic, the dinosaurs began to emerge, and it is widely believed that Welwitschia came into existence around this time.
They emerged first, as we know from fossil evidence, around about 200 million years ago. They found Welwitschia fossils, Brazil, around about 150, 160-year-old deposits. They recognized the flowers, they recognized the codes, and they recognized also some of the pollen that is in that deposit.
Today, Welwitschia is found only in the Namib Desert. The Brazilian fossils, however, are evidence of the plant's primordial existence, first growing in a time prior to the existence of the Atlantic Ocean. South America and Africa were once connected in a vast continental area known as Gondwanaland. Around 130 million years ago, the gigantic landmass gradually broke up to form the Atlantic Ocean, separating populations of plants and animals. Here in Namibia, in this now extinct supervolcano known as Mesen Crater, any large and thus presumably ancient specimens of Welwitschia can be found. Lichens cling to this giant, which stands almost 6 feet tall. Notoriously difficult to date, no one truly knows the age of any Welwitschia specimens, although scientists have estimated that a plant this size could be over 2,000 years old. Clearly, the adaptations of this extraordinary plant species have served it well throughout the ages.
So that is what we have for our gymnosperms. So we'll take a look at some examples on Thursday and walk through some stories identifying these things. There are a couple terms that I pulled out for this. And as we have our lecture quiz coming out, one of the things that I want to make sure that you have enough practice on is looking at phylogenies. So I have a link here to a phylogeny kind of practice document. It has figures that go along with it. So you'll see that this question is referring to figure 1. So you'll find figure 1 over here. Okay. So as you're studying, I'd recommend going through some of these questions, looking at the figures, making sure that you can answer them, and then you can check yourself on this key. It should be like, I have to press enter. Yeah, okay. So also, I won't click into it now so that you can do these yourself first, but that should get you started with some of the topics that are going to be covered on our quiz next week. Okay. Sound good? All right, cool. See you all Thursday then.